I want to talk a little bit more about viruses. Actually, a good bit more about viruses. I want to kind of go over the retroviral mechanism of affecting cells. This is a retrovirus. A retrovirus is just another name for a virus that contains RNA. Are you with me, Sarah?
Yes.
So this is a virus that has RNA inside of it. And it's not only going to have RNA inside of it, it's going to have an enzyme called reverse transcriptase. We're on page 369. And this enzyme is this little blue bead in this picture.
What section is this?
Page 369, 20.1. And so here's a virus connecting to a cell. And as you see, the way viruses get into cells is they latch to a protein receptor that's on the surface. That's a little protein on the surface of the cell. And here's the virus latching onto it. Now in this picture, they just show that protein right there, but actually the entire cell is covered with these proteins. So if the virus bumps into it, it's likely to latch onto it. And so once it attaches, the cell will engulf the virus and take it in, and the protein, the capsid, is injected into the cell.
Where does it get the envelope from?
I'm going to show you.
But it changes RNA to DNA or DNA to RNA.
Hold on, we haven't got that far yet. Now we get to the part where the RNA of the virus is released from the virus, and now it's just floating around in the cytoplasm. Y'all see right here? And there's the little reverse transcriptase enzyme. And what that's going to do is it's going to copy the RNA and make a DNA copy out of it. That's the opposite of transcription. Transcription is taking DNA and making an RNA copy. Remember that? So reverse transcription is going backwards, taking the RNA and making a DNA copy. And so what it does is it makes this DNA copy, which is called cDNA, or complementary DNA. It's DNA that's complementary to the RNA strand that was injected. Are y'all with me? Some of you are looking at your hair or your fingernails. It's probably time to start paying attention up here. Now, this complementary DNA strand is going to be copied again into a full DNA strand. A double-stranded DNA that enters the nucleus of the cell. So now we have the virus is no longer RNA, it's now DNA. And it's entering our cell nucleus. And inside the nucleus, it ties itself into the host cell's chromosomes.
What is the pink stuff? Not that pink stuff.
This?
The orange stuff, I guess.
Here?
No, the other stuff. Where everything went on until it went on the nucleus.
The bubbly thing?
That's
the viral DNA, isn't it?
This right here?
Yeah.
That's the cytoplasm of the cell.
Okay.
That's outside the nucleus. So here's the nucleus, here's the cytoplasm, and here's the cell membrane.
So everything takes out on the cytoplasm.
Yes. All of this takes place in the cytoplasm. The double-stranded DNA then goes into the nucleus and ties itself into the host cell chromosome. Now it's a prophage or a provirus. That's what we call it when it's in an animal cell. It's called a provirus.
Ties itself into what?
Into the host cell's chromosomes. Look up here, Ikeda. You see, this is the DNA of the host cell.
Yeah.
And now there's a piece of virus in it.
Okay. And then it leaves.
And then it'll just sit there for your whole life. And your cell, being stupid, reads the viral DNA just like it would read your normal DNA. And it transcribes it and translates it. And so here's the transcription process. Guess what these little things that look like snakes are?
Virus.
Not DNA, because it's transcription.
Viral RNA.
Those are RNAs that was transcribed from the DNA. And they will leave the nucleus, and they will go to a ribosome. These are a bunch of ribosomes here. And the RNA will go through the ribosomes and eventually make proteins. These are a bunch of proteins that are made by the ribosomes. In the endoplasmic reticulum, you get a bunch of proteins that are for the virus, you see. And then this is all put together in the cytoplasm. And see all these little proteins for the virus get put onto the membrane here. And then the virus will bud off and you have a new virus.
So what's maturation?
Cole, you're asking that envelope. That was originally part of the cell here. You see?
Oh wait, so the virus puts all those more receptor proteins so it's easier to invade it?
The virus puts these proteins on it. Those proteins are going to be necessary for getting into the next cell.
So it's kind of like wearing almost like a disguise.
See, it's see these brown proteins here?
Okay.
Those are needed for the
Okay,
virus.
so they put them there just to use it for the envelope.
Absolutely.
Where do they get them from?
They make them in the ribosomes. They're coded for on the DNA here. The DNA codes for making those proteins. All of these proteins are in the code of the virus.
So they go out and latch onto the outside of the cell?
That's right.
What's the point of latching onto that cell if it's already been infected? How does the other
Because it's in this cell that it can make new viruses.
Are there viruses?
Only that cell?
Well, they attach to
In any cell like this.
Oh, okay, I get it.
Each of these viruses can go out and infect another cell.
Okay.
Yes, it can.
So what happens in maturation?
All these proteins that are made are put together to make new viruses.
So formation of leave a new cell?
Say that again?
Like it forms a new cell to leave?
Not a cell, a virus.
Proteins make.
The proteins come together to make the virus.
Okay, and what happens in like what attaches?
See this virus up here?
Yeah.
It attaches to the cell during the attachment phase.
Okay. And then comes in.
That's right.
There's secretion and then.
I tell you what, watch this video first. Okay? I want everybody to watch it. Don't just sit there staring at your desk. This shows the whole thing, and it might make more sense to you. This is how, in particular, this is how HIV works, because HIV is also a retrovirus. Okay?
And what classifies retrovirus? The whole list
A virus that has RNA instead of DNA.
Okay.
And it does the reverse transcription.
It does reverse transcription. So again, y'all watch this. Don't be writing right now. Okay? Watch this.
Watch that.
HIV, the human immunodeficiency virus, is a retrovirus. An HIV particle attaches to the host cell membrane by way of the membrane protein CD4. The cell removes the viral coat as it takes up the virus by endocytosis. Using reverse transcriptase brought in by the virus, the viral RNA makes a complementary cDNA. The viral RNA degrades. The cDNA is the template for a second DNA strand. The double-stranded DNA enters the host cell's nucleus and integrates into a chromosome as a provirus. Eventually, transcription produces viral RNA. The viral RNA travels to the host cell's cytoplasm, where some is reserved for new viral genomes and some is translated, producing new viral proteins. These proteins are assembled to form new capsids and envelopes. The assembled retroviruses bud from the plasma membrane. The new viruses can parasitize more host cells.
So does the viral DNA stay attached to the cell DNA and keep making it, or does it?
Yes, that's the insidious part. The viral DNA is in the nucleus here, and it can literally make millions of new viruses.
It'll keep going until the cell just dies.
That's right.
It's different than regular whatever.
Like
So it kind of kills
the
this.
DNA goes and infects it?
Normally, a regular DNA virus would just have the DNA go right into the nucleus that it injects. An RNA virus injects RNA, and reverse transcriptase changes the RNA to DNA. And then the DNA goes into the nucleus. And the difference being is that RNA can mutate very easily, you see. So it's an RNA virus, so it has the advantage of being able to mutate quickly. But in converting it to DNA, it also has the advantage that it can bind itself into the host cell's chromosomes and exist there as a provirus, using the cell's machinery for the rest of the cell's life.
So normally DNA would just go straight into it and be in the nucleus, and then it just like make RNA from there. And then this one, the RNA goes in, makes DNA, and then DNA makes RNA.
That's correct.
Does it
Now you already knew that DNA made RNA, right? That was
what?
You already knew that DNA makes RNA.
Yeah.
That's the normal process.
I didn't realize that. Never mind.
Are you sure? Okay. Yeah. EKF.
Yeah. What's the name of that protein in a lot of
I'm sure you don't even know. Oh wait.
Okay. This is actually an important protein. It's called a CD4. This is the protein. Now this protein is important because one medication that stops AIDS is one of the AIDS medications is a drug that you take that has a shape that fills that spot there.
That's awesome.
And so if you have AIDS, you take this drug and it comes and it sets down right there and it's in the way, and now this virus can't bind, you see, and it can't get into the cell. And so that stops the virus. And so you don't develop AIDS.
It doesn't cure it though.
It doesn't cure it, but it stops it from making more.
Why doesn't it cure it?
Well, because these things are still floating around.
That's really, really cool.
Okay,
what's
so
the
you
difference
take
between-
it continuously though, right?
Yep.
All right.
How do
You
you-
said that it says that HIV can only last for like 9 to 11 years, right? So if
That's
we were
before
taking-
you die.
That's before it kills so many of your cells that you can no longer fight infections.
Oh.
Because this cell that it's infecting is one of your immune system cells.
Okay, so how long can HIV survive without, spreading?
Probably depends on a lot.
10, 12 years, it depends on the person.
So if you take the medicine for 10 or 12 years,
It's still in you, but the numbers are very low.
Yeah.
People live. Magic Johnson's been alive now 20 years with HIV, being HIV positive, and he still looks good. So the medications help. That's just one medication. I'll show you some other.
Okay, I know that it got the envelope from another cell, but where did like the first virus that infected it get it from?
We don't know.
Oh really?
We don't know. Nobody knows. That's a good question.
Does it kill that cell, or like, why does it make people sick?
Well, what happens is this cell is an immune cell. And what can happen is mutations of this virus will happen, and normally it doesn't hurt the cell when this virus buds off, it doesn't hurt the cell. But some mutations will eventually build up where when it buds off, the cell actually breaks open and dies. And what you end up doing is you lose a lot of your immune cells because of it. We're going to talk more about this when we do the immune system, and we'll kind of revisit this. But what eventually happens is these cells that are producing these viruses will eventually die after producing a lot of viruses, and you need those cells to be functioning to live. So you die from not having immune cells.
Like you
if
catch a
if
cold
you
and
have
die.
HIV. I think Steph was next.
Yeah, when we learned about transcription, I thought the whole point of that was because the DNA strands couldn't fit into the nuclear pores. How does that work here? Do you see what
It's a
I'm
good
saying?
question.
How does the DNA go through the nucleus?
I don't know.
Maybe it's just so small, like that doesn't look very big because viral genes aren't that long, are they?
Maybe it can fit through, but since this is so short, it's able to go through, and since a chromosome is so long, it's not able to go through. I don't know. That's a good question.
Are those-
I don't know.
Doesn't it coil up inside of there so it doesn't leave?
Are those circles nuclear pores?
Yeah, those circles are nuclear pores. Doesn't what coil up in there?
Like, isn't it coiled up in chromosomes, like inside of the nucleus, so then,
Yes.
like, it wouldn't be able to leave?
Well, it's-
No, it wouldn't be able to leave now, but she's asking how it gets in there,
It's not coiled.
because it's double
But
stranded.
even when the
Not single
other
stranded.
DNA was not coiled up, it still couldn't go. That's the whole point of changing it into an RNA.
before it left the
Yeah, I'm not sure still. Somehow it gets in there though. Yes?
What's the name of that thing that looks like a U that's transcript? No.
That's
That thing?
Yeah, what's that called?
That's called DNA polymerase. It's an enzyme that copies one strand of DNA into another. It's the same enzyme when you're copying DNA that'll copy another strand of DNA. Yeah.
Okay. Also, since the virus doesn't use the reverse transcriptase in its own like mechanics, do we know where it came from, or is that kind of a mystery too?
It came from the virus.
I mean, like
that's a good question. Like evolutionarily, where it came from. I don't know if they know that either, but I would guess that it's probably a derivative of.
So like it took it
of RNA polymerase that actually does.
So like it took it in from a host cell or something.
Maybe, yeah. Most they think the origin of most viruses is from cells.
Mm-hmm.
That when a cell dies and comes apart, if it has the right proteins and the right DNA and the right configuration, it'll become a virus.
Have they ever tried that, like trying to make a virus?
I don't know. I don't know.
It'd be easier than making a cell.
Yes.
Right?
If viruses respond to certain receptors like CD4, why don't people create, like, I don't know, things like lysosomes that have only CD4 proteins on them so that the viruses will lock onto those, and thinking that it's actually just another cell, it opens up that cell and then it just eats it.
Yeah? Sounds good to me.
Why don't you just take like preventarian?
Why don't you make that? Yeah. Sounds like a good idea. I don't know why they couldn't do it. I don't know if it's easy to do. I don't know. Yeah.
Do it. It's like
Okay.
a million.
Question?
Yes. What, you know how in maturation, when the proteins are made, or whatever?
Down here?
I don't know.
Right here?
Yeah.
Okay.
Are the proteins, you know how you said it's or the video said it's like surrounded in proteins so that it can go onto other things? Are those specific for like each different cell?
Each virus.
Are the proteins on the outside, like can the virus that's made go into anything at once?
No, they're specific. In other words, the only cell that has a CD4 protein here is an immune cell. So this HIV virus can only get inside immune cells. It's specific. It only affects a specific type of cell.
Why doesn't
So even though that's HIV, for every different virus, it goes into a different immune cell?
Not necessarily an immune cell. HIV affects immune cells. The common cold affects mucous membrane cells. Herpes affects cells in the genital area and such. So different viruses affect different types of cells. Yes, Jordan.
Why wouldn't the HIV viruses leave? Like, if you stopped it from transcription and like even go and attaching to the cell, why would it still be around if like why wouldn't it just leave the body?
Well, it could leave the body if you were cut or something like that, but it just kind of circulates inside the cells.
your immune system fight them?
Your immune system can fight them. The drugs aren't perfect. Even if you take a drug that blocks this receptor, the drug won't necessarily block every receptor. There's thousands of receptors on each cell, and there are thousands of cells. So you can't be assured of covering up every receptor with the drug.
I mean,
there are also side effects of the drugs. Like, this receptor has a reason for being there too, and there might be some reason for that it needs to take in other things, and it can't do that if the drug's on it. So there are side effects that come with drugs. It's blocking the virus from getting in, but it might be blocking something else important from getting in too, and that could have a bad effect. So these medications aren't perfect. Yeah.
What would happen if another virus got into a cell that already had a virus?
That can happen. And if that happens, often the genetics of the viruses can mix
And create a new one.
and create a whole new virus that has different properties from two
So
different
it's kind of like
viruses.
mix?
Yeah.
Yeah.
And that often happens, and they're scared, for instance, of different types of flus. Like the reason why swine flu, if people got the swine flu that mutated from a pig in their body, and then got another type of human flu that's easily transmissible between people, and the swine flu mixes with the human flu inside one of their cells, then you could get this terrible flu that normally you might get from handling poultry or pigs, and it'd also be able to be passed from person to person. That could start an epidemic.
So it become like...
Yeah, if it got some mutation by mixing with the normal flu that was airborne, and it had the bad sickness genes from this other flu that's not airborne, then now you can get a new flu that's airborne and really bad. And so that sort of thing does happen.
Was the bubonic plague a virus?
No, that was a bacterium. We'll talk about bacteria next.
Okay, I have another question. So how exactly have they not found HIV rates again?
It's hard. Some of the drugs help, but to cure it it constantly mutates.
So in order to...
So it might mutate where it has a little bit different shape and can get into the cell a different way or something like that, and now you don't, your drug doesn't work anymore. So there's mutations that you have to worry about.
So would the way we could help create a cure for it, if there was, would we have to know who the host was? Because isn't the host typically, the very first
Yeah, in
patient?
outbreak, they have to find them.
Yeah, that's possible. I mean, I don't know. Each virus is different. The way you treat them is different. Look, watch this videos about some of the treatments for HIV.
At present, treatment for HIV infection involves using a combination of drugs. One type of drug inhibits nucleic acid replication. For example, when AZT is incorporated into DNA by reverse transcriptase, no additional nucleotides can be added. A second class of drugs.
Okay, hold on now. So AZT, have you ever heard of that drug? That was the first drug for HIV. Here's the viral RNA. Here's reverse transcriptase. What's the reverse transcriptase going to do? What's its job here?
RNA.
It changes the RNA into DNA. And so it's going to attach and start making DNA. And these are DNA nucleotides. And see, it's a T and a C. Well, AZT is a drug that is shaped like a nucleotide. And so this AZT, when it sticks to this, when it binds, the reverse transcriptase puts it on thinking it's a nucleotide, but it's not. It has a little bit different here, a little bit different molecule, and it keeps new nucleotides from being able to be added. So reverse transcriptase cannot do its job. And so that stops the virus. That's one of the drugs.
Nucleotides can be added. A second class of drugs inhibits a protease involved in cleaving large polyproteins into functional segments. When the protease is inhibited, virus particles cannot be produced. Use of a combination of drugs directed at different targets reduces the likelihood of the development of resistance by mutation. Research is currently underway to develop new treatments for HIV. Chemicals called chemokines appear to inhibit HIV infection by binding to and blocking the CXCR4 and CCR5 co-receptors on host cells. A search is in progress to apply such chemicals in HIV treatment. Some individuals who are HIV positive but did not develop AIDS have a mutation in the CCR5 receptor that makes it defective. Research is in progress to determine whether disabling the CCR5 receptor might be effective in controlling AIDS in infected patients. An antiviral factor called CAF is found in CD8 cells. Research is in progress to determine if this substance could be used to block replication of HIV. There is currently no approved vaccine against HIV infection. One problem is that HIV surface proteins such as GP120 mutate at a high rate. Scientists may be able to construct a vaccine using an HIV with a defective form of the viral gene NEF. Viruses with the defective NEF gene seem to have reduced reproductive capability. allowing the immune system to keep the virus in check.
Here's another thing about antiviral agents, things that work against viruses. This is all new science.
Viruses are difficult targets for selective treatment because they rely to a large extent on the host cell's metabolic activities for their replication. Most antiviral agents inhibit viral uncoating, interfere with viral nucleic acid biosynthesis, or prevent the assembly and release of viral particles. After the virus enters a host cell, the protein coat must be removed from the nucleic acid in order for replication of the nucleic acid to occur. Amantadine blocks penetration and uncoating of the influenza A virus. Foscarnet is an analog of pyrophosphate. It binds to the active site of DNA polymerase and blocks the cleavage of pyrophosphates from nucleotides. It is used to treat cytomegalovirus and other herpes virus infections. Replication of nucleic acids involves additions of nucleotides to the free 3' end of the growing chain. Nucleoside analogs are incorporated into nucleic acids during synthesis, but then prevent further synthesis. For example, azithothymidine, AZT, an analog of thymidine, becomes phosphorylated in the cell and is then incorporated into nucleic acid by reverse transcriptase. Since the 3' position of the deoxyribose is occupied by nitrogen instead of a 3' OH, no additional nucleotides can be added. AZT has been used in treatment of retroviral infections. The HIV genome is transcribed into messenger RNA and then translated into a large polypeptide. A site-specific protease cleaves the polypeptide into functional proteins. Protease inhibitors interfere with this protease and prevent production of functional proteins. Protease inhibitors have been used to treat AIDS patients.
Let me talk about that one real quick. Okay, so here's the provirus of the HIV virus sitting in the nucleus. And that's going to be translated into proteins, right? Into messenger RNA, and then into a protein in the ribosome. There's the protein. Now this long protein actually has to be cut. It has to be cut there and there. And when it's cut there and there by a special enzyme called a protease, it becomes the 3 proteins, the yellow, green, and blue protein here, that are needed for the virus. So we got to cut this long thing up for the virus to work. So what you could do is you could give a drug to the person that keeps the protease from working. It's a drug that binds to the active site of this enzyme and keeps it from being able to cut this protein. That's an AIDS drug. It's called, there it is, a protease inhibitor. And it binds to the protease, and now the protease can't cut.
To treat AIDS patients. Some interferons are antiviral. These proteins are produced by an infected host cell.
We'll talk about interferons at a later time.
I know the difference between, I know that HIV turns into AIDS.
Uh-huh.
You know what?
Yes.
What's the difference?
When HIV is a virus. Emery, you getting this? You don't look too good. HIV is a virus, and when enough of the immune cells die, the disease is called AIDS. So if you have AIDS, you're lacking immune cells. Okay? If you have HIV, you just have the virus in you. You still have immune cells.
So it's just after the virus has killed enough cells, then it's considered AIDS.
Then you have AIDS.
You catch the cold and die, right?
It worked out this morning, and
Right.
it increased our immune system.
Oh, really?
Yeah.
Well,
we're
congratulations.
We're not going to get it. Made me tired.
Don't people, isn't
That's
that
why
a
we don't
disease
like it.
that people are immune to HIV?
Yes, that's what they were just showing. Some people are immune to HIV because they have a mutation in the protein that the HIV latches onto to get in the cell. It's not shaped right. So they're immune to AIDS.
Yeah. Then can the immune system kill that virus even though it's not doing anything? Or is it? That's kind of what I'd like to know.
Yeah. Maybe it can. I don't know how that works. Okay.
So we all still more to learn, still.
That's right. I don't know it all. There's an entire field. You can become a virologist and study this, not just for 1-2 days, but for 4 years, and to get to understand all there is to know. Yeah. And they're learning new stuff all the time. Now, I want to get into the next section about prokaryotes. And prokaryotes are very tiny organisms. Let me show you the size. Look at the size. Now, this is an electron micrograph of the tip of a needle, like a sewing needle.
That's insane.
And in actuality, it's not too sharp when you're looking at it under an electron microscope. Every little orange dot you see is a bacterium. Okay? So this is how small these things are. What they did was they took a needle and stuck it in some bacteria that were growing on a dish, and then took a shot of it. So that's what the bacteria look like in size. Now you can imagine, if you want to understand how big a virus is, you could probably fit 500 or 600 viruses in one bacterium. So they're small. Viruses are small. And bacterium are a lot bigger than viruses. What's that? One virus can make you sick.
When you say one, you mean one once it multiplies like a good million times, or just like one tiny little virus inside?
No, once it multiplies. See, one virus can get in one of your cells and multiply, and now you got 100 viruses. And if each of those 100 get in another cells, now you got 10,000 viruses. You're not usually sick from the virus. You're sick from all your cells dying. You see? If all your cells die, you're going to be sick.
How many cells die a day in your body?
A lot, and a lot of new ones are made, but when they're infected with viruses, they die a lot quicker.
It scares me.
So, these things are too small. Bacteria you can even barely see with our microscopes, our light microscopes, you can barely see bacteria. They're just so tiny. So, how do we know they're there at all? Pasteur did a lot of the original research that figured out that there were bacteria out there. He took a flask, and he would boil the flask and put a cork on top and seal the flask, and the flask will never spoil. If the broth here, this is broth like soup, you see, and if you boiled it and then put a cap on it, it'll never spoil, even after years and years, because it's been sterilized, you see. This is what canning foods is all about. You put the food in a can, you seal the top, then you heat it up, kills all the bacteria inside, and the food will stay good for years that way.
Is that like mason jars?
Like mason jars, yeah. Absolutely.
But then why do they have expiration dates on them though?
Well, that's not completely pasteurized like milk. Sometimes.
Yeah, things with expiration dates will spoil, but
like you can super pasteurize milk, but that just.
There might be chemicals in it that will break down over time, and that might be why they have the expiration dates. But they won't spoil. Now, so what Pasteur did was he took this flask and he'd take it outside and he'd open it just briefly, and he'd close it again, and guess what? Flask would spoil. So he figured out that there must be something floating around in the air that when you open it, it goes down in there and starts multiplying. And that's what spoiling is, multiplying bacteria.
What?
So he found out that if you go outside and open the cap, it spoils 89% of the broths. But 89% of the flask that he took out there would spoil if you went outside and opened it. If you opened it inside, only 32% would spoil. So there's more bacteria floating around outside than there is inside. Hold on, Cole, for just a second. Furthermore, he took this flask. Y'all need to be looking at this. This flask has what is called a swan neck flask. It's kind of shaped like an S. And he boiled the broth here. And because it's shaped like an S here, that never spoiled. And the bacteria, if they came in through this opening here, they would just fall to the bottom and see they're too heavy to go up this way against gravity and fall in there. So this S, the swan-shaped flask, never spoiled. But if you tip the flask over so that the broth runs down the swan neck, and then you tip it back, it'll spoil. And so a lot of his research proved that there was something floating around in the air that you couldn't see that was spoiling his broth. And there's still in a French museum one of Pasteur's original swan neck flasks sitting there unspoiled.
That's awesome.
After 200 years.
I don't know what it tastes like.
Isn't that cool? There's some broth in a flask, and it's open to the air, but it's not spoiled yet. So that's some pretty neat experimentation. Yeah, Cole, what were you going to ask?
I was just wondering how we didn't know it wasn't just like a chemical reaction.
Well, chemical reaction with what?
Like with just something in the air, like oxygen.
Well, should have still been oxygen here even after you boiled it.
Okay.
And that shouldn't be, it must be something that's killed by boiling. So, he just kind of concluded there's probably something, and it turns out that those are bacteria.
Okay.
This is what a bacterium looks like. It's got a cell wall on the outside of it. It's got DNA coiled up in the middle. Doesn't have a nucleus.
No.
It's got ribosomes. But the ribosomes of bacteria, of prokaryotes, are a little bit different than our ribosomes in our cells. They're a little bit smaller. They aren't quite arranged the same. It's got a plasma membrane under its cell wall. It's got cytoplasm. I want you to start reading 20.2. I want you to read all of 20.2. I didn't get done with it.
Did you say it doesn't have a nucleus?
Does not have a nucleus. It just has a nucleoid, which is DNA kind of floating in the center of the cell there.